Millions of Americans spit into a tube, sealed it in a box, and mailed it off without a second thought. Each one had already checked a box on a form somewhere, a box that said white, simple, certain, unquestioned. Then the results came back, and for nearly 1 in 20 of them, the story their DNA told was not the story they had always believed about themselves. Buried inside their genetic code was African ancestry they never knew existed. Not a rumor, not a family legend, whispered at Thanksgiving. Not a guess from a distant cousin doing genealogy research on a weekend. Actual, measurable, mathematically calculated African DNA, sitting quietly in their chromosomes for generations, inherited without their knowledge, passed down silently through parents and grandparents who may themselves have had no idea it was there, waiting patiently to be found. And in some southern states, that number did not sit at 1 in 20. It climbed all the way to 1 in 8. This is not a rare glitch in a lab. It is not a fluke result from one strange test kit. It is a pattern, and it shows up again and again in study after study across tens of thousands of people. When scientists lined up the numbers, state by state, family by family, they found something that historians had been arguing for decades, but had never been able to prove with hard physical evidence. Race, as Americans have understood it for centuries, does not match the biology sitting inside their own cells. And the gap between the story people tell about their ancestry and the story written in their genome turned out to be so consistent, so geographically patterned, that it reads less like random noise and more like a fingerprint left behind by history itself. The research that uncovered this did not come from a single, isolated experiment. It came from one of the largest genetic ancestry studies ever conducted in the United States, a collaboration between population geneticists and the genetic testing company 23andMe. Researchers gathered DNA samples from more than 160,000 people, most of them customers who had sent in a cheek swab or a vial of saliva, hoping to learn a little more about where their families came from. Alongside the DNA, participants filled out detailed questionnaires. They were asked how they identified racially. They were asked where their ancestors were believed to have come from. They were asked about their family history, their geography, their sense of self. Then the scientists did something no one had done at this scale before. They compared what people believed about themselves to what their DNA actually showed. The comparison relied on a method geneticists call ancestry informative markers. These are specific points scattered across the human genome where the genetic code tends to differ in consistent, measurable ways between populations that were geographically separated for long stretches of human history. No single marker proves anything on its own. But when researchers look at hundreds of thousands of these markers together, patterns emerge. A person's DNA can be broken down into percentages. This fraction inherited from ancestors in West Africa, this fraction from ancestors in Western Europe, this fraction from indigenous American populations, and so on. It is not guesswork. It is statistics. Applied to biology, refined over more than a decade of research into human population genetics, and it has become precise enough that testing companies can now estimate ancestry proportions down to fractions of a percent. When the team ran the numbers on the self-identified white participants in their sample, the overall figure came out to just under 4% carrying detectable African ancestry. On its own, that might sound small, 4 out of every 100 people. But 4% of a country with hundreds of millions of people is not a rounding error. It represents millions of individual human beings, alive today, walking around with a piece of ancestry hidden inside them that contradicts the racial identity they have carried their entire lives. And the number was never evenly spread. It clustered. It concentrated in specific places, and when the researchers mapped those places, the map told its own story. South Carolina and Louisiana sat at the top of the list. In those states, the rate of detectable African ancestry among people who identified as white climbed as high as 12%. That means in some communities, roughly 1 out of every 8 people who checked the box for white on a census form or a job application or a college enrollment sheet carried genetic ancestry that would have placed them in an entirely different, legally defined racial category under the laws that govern those same states for most of American history. Georgia, Alabama. Other parts of the Deep South showed elevated numbers as well, hovering close to 1 in 10. Mississippi and parts of North Carolina followed a similar pattern, each region carrying its own slightly different percentage depending on local demographic history, the size of the enslaved population that once lived there, and how much migration in or out of the state occurred over the following century and a half. Meanwhile, in states further north and west, the numbers dropped off substantially, sometimes falling below 1%. This was not random distribution. This was geography acting as a kind of historical record, preserved not in books or archives, but inside the double helix of ordinary people's cells. To understand why the numbers cluster exactly where they do, you have to go back to the specific mechanics of American slavery and the century of legal segregation that followed it. The Deep South held the largest enslaved populations in the country for the longest period of time. Forced sexual relationships between enslaved women and white slaveholders were common and often violent, a brutal and well-documented feature of plantation life. Children born from those unions were, under the laws of the time, still considered black, regardless of how much European ancestry they carried, because Southern law defined race based on any known African ancestry at all, a principle that later became infamous as the one-drop rule. That rule was never really about biology. It was a legal and social tool built to preserve a rigid racial hierarchy, to make sure that mixed ancestry could never dilute the boundary separating who was enslaved from who was free, and later, who held full citizenship rights from who did not. But biology does not obey legal categories, and DNA does not check boxes on a form. Over the following generations, some descendants of those unions, particularly individuals with lighter skin, migrated, married, and quietly folded themselves into white communities, a practice historians call passing. It happened during Reconstruction. It happened during the Jim Crow era, when being classified as black carried the risk of violence, economic exclusion, and legal discrimination that shaped every aspect of daily life. It happened during the Great Migration, when millions of black Americans moved north and west, and some family lines, especially those with ambiguous or lighter appearances, drifted across the color line entirely, sometimes deliberately, sometimes simply through the passage of time and the fading of family memory. Names changed, records were altered or lost, family stories were reshaped, softened, or erased outright, sometimes to protect the people involved, sometimes out of shame imposed by a society that punished any acknowledgement of black ancestry. Over enough generations, the genetic trace remained even as the memory of it disappeared. A person today filling out a DNA test might have absolutely no oral history connecting them to black ancestors. No family story, no old photograph, no hint passed down at the dinner table. And yet the DNA remembers what the family forgot. This is precisely what the researchers found when they cross-referenced ancestry percentages against self-reported family narratives. Many participants with detectable African ancestry had no idea it was there. Their family trees, at least the version they had been told, contained no hint of it at all. There are countless individual stories tucked inside those aggregate numbers, the kind of stories genealogists encounter again and again once they start pulling on a genetic thread. A person grows up in a small town in the Carolinas, raised entirely within a white family, attending a white church, absorbing a family history that traces cleanly back to European immigrants who arrived generations ago. Then a DNA test arrives in the mail, and buried in the results is a segment of African ancestry too large to be coincidence, too consistent to be measurement error. Sometimes a little digging through old county records turns up the answer, a great-great-grandmother, whose racial identity shifted somewhere along the way, a name quietly dropped from a family Bible, a birth certificate that does not quite match the story everyone assumed was true. Sometimes no paper trail survives at all, and the DNA becomes the only remaining evidence that a piece of the family's past was deliberately, carefully erased. Claudio Sant, a historian at the University of Georgia who studies exactly this kind of hidden demographic history, put it plainly when the findings were published. Historians, he said, have spent a generation writing books arguing that race is not a biological fact, but a social and cultural construction, something built by law, custom, and power, rather than something rooted purely in ancestry. What this genetic research did was take that argument and hand it a completely different kind of proof. Instead of legal, documents or census records or plantation ledgers, it used DNA itself, the most intimate and supposedly objective evidence a person can offer, and showed that the categories built around it were leaking at the seams the entire time. It would be easy to stop the story there, but the same massive dataset revealed something just as striking on the other side of the color line. Researchers also examined the genetic ancestry of people who identified as African American, and what they found mirrored the same complicated history from the opposite direction. On average, the DNA of self-identified African Americans in the study showed substantial European ancestry, typically somewhere between 20 and 25%, though the range varied enormously from person to person and again by geography. Some individuals carried barely any detectable European ancestry at all. Others carried well over half. The Deep South again showed patterns tied directly to the history of slavery, while other regions reflected different waves of migration, marriage, and mixing that occurred over the past 3 centuries. Latino participants in the same study showed yet another layered pattern, a genetic mixture of European, Indigenous American, and African ancestry in proportions that varied by country of origin and by region within the United States, reflecting the distinct colonial and migration histories of Latin America, from Spanish colonization to the transatlantic slave trade's reach into the Caribbean and South America, to indigenous populations that predate European contact entirely. A participant with roots in the Caribbean might show a substantial African ancestry component, alongside European ancestry from Spanish colonization, while a participant with roots in parts of Mexico or Central America might show a heavier indigenous American component instead, and someone from Argentina or Chile might lean far more heavily European, reflecting very different immigration waves. Taken together, the three groups painted a picture of a continent and a country whose population has never actually existed in the neatly separated racial boxes that census forms and cultural habits have long assumed. It is worth pausing on just how large and methodologically careful this particular study actually was, because the scale is part of why the findings carry so much weight. This was not a small pilot project with a few hundred volunteers. It drew on one of the largest consumer genetic datasets ever assembled at the time, cross-checked against self-reported family history spanning all 50 states, and published in a peer-reviewed scientific journal where other geneticists could scrutinize the methodology line by line. The researchers controlled for factors like sample size, differences between states, adjusted for how ancestry informative markers behave across different reference populations, and cross-validated their ancestry estimates against known pedigrees and previously published population genetics research. That level of rigor is precisely why historians and geneticists alike treated the results as more than just an interesting anecdote. It was, for the first time, hard population-scale genetic evidence for a pattern that oral history, old legal records, and fragmented family stories had hinted at for generations, but had never been able to quantify with this kind of precision. None of this is really surprising once you step back and look at the deeper biological story running underneath American history, because the patterns found in this study are really just a localized, recent chapter of a much older and much larger story about human genetic diversity as a whole. To understand that bigger picture, you have to go back tens of thousands of years, long before the United States existed, long before slavery, long before Europe or Asia or the Americas were populated by modern humans at all. Homo sapiens originated in Africa. That much is settled, supported by fossil evidence, archaeological evidence, and an overwhelming convergence of genetic evidence collected over the past several decades. For most of the history of our species, every single human being alive lived on the African continent. Populations there had tens of thousands of years to grow, split apart, migrate internally, and accumulate genetic variation across an enormous and ecologically diverse landmass. Then, sometime around 60,000 years ago, give or take depending on which estimate you use, a relatively small group of humans left Africa and began spreading into the Middle East, then into Europe, Asia, Australia, and eventually, tens of thousands of years later, into the Americas. Here's the part that surprises most people the first time they hear it. Explained clearly, that migrating group was small, a tiny fraction of the total human population at the time. And because they were small, they carried with them only a fraction of the total genetic diversity that existed back on the African continent. Every population outside of Africa today, every European, every Asian, every indigenous American, every Pacific Islander descends from that relatively narrow genetic bottleneck. Population geneticists call this the founder effect. Think of it like a massive, diverse library with millions of unique books representing thousands of generations of accumulated variation, and only a small handful of those books being carried out the door to start a new library somewhere else. The new library can grow enormous over time. It can develop entirely. New characteristics, new local adaptations, new patterns shaped by different environments, different diets, different climates. But it will never contain the full original diversity of the collection it came from, unless new material is brought back in. This single fact explains something that continues to surprise people every time genetic diversity is actually measured directly. Two random individuals sampled from neighboring villages within a single African country can, on average, carry more genetic difference between them than two random individuals sampled from opposite ends of Europe. It sounds almost backwards at first, especially given how casually the word African gets used in everyday conversation, as though it describes one uniform population. It does not. Africa contains, by a wide margin, more human genetic diversity than any other continent on Earth, a direct consequence of being the place where our species spent the overwhelming majority of its existence, accumulating variation over hundreds of thousands of years before any small subset of that population ever left. This deep, foundational reality about human genetic diversity has quietly reshaped one of the more important tools in modern medicine, a tool most people have never heard of, but that touches nearly every piece of genetic research conducted today, the human reference genome. When scientists first assembled a complete map of human DNA in the early 2000s, they needed source material, actual DNA donated by real people, to build the template that every subsequent genetic study would be compared against. Given how the history of genetics research As often unfolded, many people assume that reference template must be overwhelmingly European in origin. It is not. The human reference genome is actually a composite, stitched together from DNA donated by more than 60 different individuals. Roughly 70% of it comes from the DNA of a single donor, an African-American man from Buffalo, New York, known in scientific literature only by the code name RP11. His ancestry itself was mixed, roughly half, derived from European lineages and half from sub-Saharan African lineages, a living, walking embodiment of exactly the kind of blended genetic history this entire investigation keeps circling back to. The remaining fraction of the reference genome comes from a patchwork of other donors, including individuals of East Asian descent and several more of European descent, along with dozens of additional contributors whose ancestral backgrounds were never fully recorded or made public. In other words, the master template underlying an enormous share of modern genetic medicine, used to identify disease-linked mutations, to interpret ancestry test results, to guide pharmaceutical research, was never a clean, single ancestry blueprint to begin with. It has always been a mosaic, and one of its largest single contributions traces back to a man whose own ancestry embodied precisely the kind of mixing that the study of self-identified white and black Americans later confirmed on a massive population scale. This has real, practical consequences beyond historical curiosity or philosophical debate about the meaning of race. For years, geneticists and public health researchers have raised concerns that most large-scale genomic research databases remain heavily skewed toward participants of European ancestry, a legacy of where genetic research funding infrastructure and recruitment efforts have historically concentrated. That imbalance matters because genetic variants linked to disease risk, or to how a person metabolizes a given medication, do not distribute evenly across human populations. A variant that shows up clearly in a study population of primarily European ancestry might be rare or entirely absent in populations of African or Asian ancestry. And the reverse is just as true. Databases skewed toward one ancestral group risk missing disease-linked variants relevant to everyone else, which in turn risks producing medical tools, risk calculators, and even prescription guidelines that work well for some populations and poorly for others. Expanding the ancestral diversity of genetic databases has become one of the more urgent priorities in modern genomics, not as a symbolic gesture, but as a matter of basic scientific accuracy and medical equity. There's a second, quieter medical dimension to this story as well, one that stretches beyond ancestry percentages and into the daily practice of medicine itself. Some medical guidelines historically used race as a rough proxy for underlying genetic or physiological differences, adjusting certain diagnostic thresholds or treatment recommendations based on whether a patient was categorized as black or white on their chart. Researchers have increasingly pushed back on that practice, arguing that using a broad social category as a stand-in for biology is scientifically shaky precisely. Because of findings like these, if a meaningful percentage of people categorized as white carry African ancestry, and a meaningful percentage of people categorized as black carry substantial European ancestry, then race-based clinical shortcuts risk misclassifying real patients based on a category that does not reliably track the underlying biology those guidelines were supposedly built around in the first place. Many medical institutions have since moved toward using more direct measures, actual genetic testing or specific biomarkers, rather than relying on a patient's self-identified race as a proxy for their underlying biology. Layer all of this together: the hidden ancestry findings among self-identified white and black Americans, the deep genetic diversity patterns rooted in humanity's African origins, and the surprisingly mixed makeup of the very reference genome underlying modern genetic science, and a much larger picture starts to come into focus. Race, as it is commonly understood and used in everyday life, on census forms, in casual conversation, in decades of American law, was never actually describing a clean biological category. It was describing a social and legal categorization system, one built primarily around visible traits like skin color and enforced through custom, law, and for a large stretch of American history, outright violence. Genetics, when it is actually measured carefully, tells a messier, more tangled, and in many ways more honest story, one of constant mixing, migration, and blending that stretches back not just centuries, but tens of thousands of years. None of this means genetic ancestry is meaningless, or that population-level genetic patterns do not exist. They clearly do. Geneticists can identify broad ancestral clusters with real statistical confidence, tracing where a person's ancestors likely lived hundreds or thousands of years ago, and that information carries genuine scientific and even medical value. What the research actually challenges is something narrower and more specific: the assumption that those ancestral clusters map neatly onto the small handful of broad racial categories that societies have chosen to organize themselves around. They do not. A person's DNA can trace to West Africa, Northern Europe, and indigenous America all at once in proportions that shift dramatically from one individual to the next, even within families, even between siblings who inherited slightly different combinations of the same parental DNA. This growing body of research has also fueled the massive expansion of consumer ancestry testing over the past decade and a half. Millions of people now send in DNA samples every year, driven by curiosity, by family history projects, by a simple desire to know more about where they come from. For some, the results confirm exactly what they expected. For others, the results deliver a genuine, sometimes emotional surprise, uncovering ancestry, and occasionally entire biological relatives that family stories never mentioned. Genealogists and historians have increasingly started using this data alongside traditional archival research, cross-referencing DNA results with old records, ship manifests, plantation documents, and census data, piecing together family histories that were deliberately obscured or simply lost to time, sometimes reconnecting descendants of enslaved people with distant relatives descended from the very families that once enslaved their ancestors, a discovery that carries enormous emotional and historical weight for the people involved. Separate research, digging into a different layer of the genome, adds another dimension to this picture, one focused specifically on maternal lineage. Scientists have compared mitochondrial DNA, the small strand of genetic material passed down almost unchanged from mother to child across generations, from African American populations, against large reference databases of mitochondrial sequences collected from across sub-Saharan Africa. More than half of the African American maternal lineages sampled matched common sequences shared across multiple African ethnic groups at once, rather than tracing cleanly back to any single identifiable nation or ethnicity. Roughly 40% did not match anything in the existing African reference databases at all, a reflection of how incomplete those databases still are, and how much genetic diversity across the African continent remains undersampled even today. Fewer than 1 in 10 sequences matched a single specific ethnic group with real confidence. That finding carries its own quiet significance for descendants of enslaved people searching for a specific ancestral homeland, because it shows that the transatlantic slave trade pulled from an enormous, overlapping mixture of West and Central African populations, whose descendants were then further mixed together through generations of forced relocation, sale, and family separation within the Americas. For many African Americans, hoping a DNA test might point to one specific country or one specific ethnic group, the underlying genetic reality is often far more blended and far more complicated, a reflection of exactly how sprawling and brutal that historical displacement actually was. The scientists behind the original large-scale study were careful to frame their findings not as some kind of shocking revelation about individual people, but as a demonstration of how deeply intertwined American genetic history actually is, whether people realize it or not. The patterns they uncovered were not anomalies. They were the expected, almost mathematically predictable outcome of centuries of forced and voluntary human contact across a continent that Europeans, indigenous peoples, and enslaved and free Africans all inhabited together, sometimes in exploitation, sometimes in defiance of law, sometimes simply through the ordinary passage of generations living in close proximity to one another. The surprising part was never really that the mixing happened. Historians already knew that much. The surprising part was how consistently and how precisely that mixing could now be measured, mapped, and confirmed at the level of individual DNA molecules using tools that simply did not exist a generation earlier. Standing back from all of it, the deepest takeaway is not really about any single percentage or any single state or any single surprising family discovery uncovered through a mail-in testing kit. It is about what those numbers, added together across hundreds of thousands of people, ultimately reveal about the nature of human categorization itself. The lines societies draw around race have always been drawn with rulers made of law, custom, and power, not with the actual tools of biology. DNA, when it is finally read carefully and compared honestly across an entire population, does not respect those lines. It moves fluidly across generations, across regions, across the categories people have used to organize themselves for centuries, carrying quiet evidence of every migration, every forced separation, every hidden relationship, and every reunion that shaped the population of an entire country. The tube of saliva, sealed in a box and mailed off by an ordinary person checking a box marked white, was never just measuring one person's ancestry. It was, in its own small way, holding up a mirror to the entire tangled, complicated, and deeply human history of an entire nation, one cell at a time. That mirror does not flatter anyone, and it does not condemn anyone either. It simply reflects what actually happened, generation after generation, on plantations and in cities, in wartime and in peacetime, in relationships built on love and in relationships built on force, in families that stayed together and families torn apart by sale, migration, or fear. Every one of those events left a trace, encoded not in ink, but in nucleotides, passed silently from parent to child for hundreds of years until modern science finally learned how to read it. The numbers on a page, 4% here, 12% there, 20% on the other side of the ledger, can look abstract at first glance. But each fraction represents a real ancestor, a real decision, a real moment in history that rippled forward far longer than anyone involved could have imagined, eventually arriving in a small plastic tube on a stranger's kitchen counter waiting to be decoded.